Good morning, my dear students. Today we are here to discuss the porphyrin synthesis. You must remember porphyrin synthesis is a very important topic because through this we are going to study the heme synthesis. And heme, as you know, is very important substance of our body and it is involved in many important substrate formations. So let's begin. Let's first start with the introduction to porphyrins. Porphyrins, as you all know, are a group of heterocyclic macrocycle colored organic molecules. They are composed of four pyrrole subunits interconnected at the alpha carbon by methane bridges. They are necessary for synthesis of heme-containing compound, most important being hemoglobin. Then comes myoglobin, and then various enzymes like cytochromes, catalases, peroxidases, and tryptophan pyrrolase. So when you talk about heme synthesis or porphyrin synthesis, just do not limit yourself by saying hemoglobin and myoglobin. You have other enzymes that are synthesized in your body, which are important for the functioning of the body. And porphyrinogen are the precursors. You must remember there is an inactive form, which are the precursor forms. We call them as porphyrinogen, which when they get exposed to the ultraviolet light, they become reactive, they become active, and they are form porphyrins. And these porphyrins are the ones which are responsible for most of the symptoms of porphyria, especially the one which is sensitive to light.
So let us start with synthesis of porphyrins. Whenever you study synthesis of any compound, you have to understand about few things. The first and the foremost should be where is it being synthesized. So looking at the site of synthesis of porphyrin, there are two major sites for synthesis of porphyrins, liver, hepatic corresponding to 80%. The synthesis in liver is variable, which indicates that liver is under regulation. So there are enzymes which can be induced and there are enzymes which can be inhibited, but only in the liver. The other site of synthesis is bone marrow or the erythropoietic synthesis, and this corresponds to 20% of the porphyrin synthesized in the body. This synthesis, you must remember, is always constant. So that means it cannot be turned on or off and it is always expressed. Then the starting materials. We just require two starting materials for porphyrin synthesis. One is the simplest amino acid glycine and the second is succinyl-CoA. Succinyl-CoA, the source of succinyl-CoA is from the TCA cycle. Once we have these materials, then we need to know how many steps do we need to synthesize porphyrins. We need a total of 8 steps for synthesis. And mind it, any cell which does not have mitochondria, for example, the RBCs, the mature RBCs, they do not have mitochondria, they will not be able to synthesize the porphyrins. So porphyrin synthesis takes place in both the mitochondria and the cytoplasm. Step number 1, 2, 6, 7 and 8 are, are performed in the mitochondria, and step 3, 4 and 5 are performed in the cytoplasm. So there is shuffling of porphyrin during the synthesis from mitochondria to cytoplasm and then finally back into the mitochondria. Then we need to know about the rate limiting step. The rate limiting step is the step where there is the requirement of the enzyme delta ALA synthase. And this is the first step of the porphyrin synthesis. And this step and this enzyme is the enzyme which regulates the cycle. So it is mainly the delta ALA synthase from the liver which undergoes regulation and regulates the porphyrin cycle. Okay. Feedback. Now feedback inhibition is by the inhibition of the enzyme delta ALA synthase, which is the hepatic enzyme. And as we go through the process, we will require that the final product, which is the heme, is the product which is responsible for inhibiting the delta ALA synthase enzyme. So let us start with the synthesis of porphyrin. Step 1 occurs in mitochondria and we have the two substrates with us, which are the glycine and succinyl-CoA. These two molecules, glycine and succinyl-CoA, undergo a condensation reaction and in presence of the enzyme delta ALAS synthase, which is also dependent upon the pyridoxal phosphate. So any person who will be having a deficiency of pyridoxal phosphate is more likely to have inhibited or a slowed heme synthesis and might have anemia. So that's the basis how pyridoxal phosphate leads to anemia. So in presence of delta ALA synthase in the mitochondria, glycine and succinyl-CoA combine to form a product which is known as alpha amino beta keto adipic acid. Now remember that alpha amino beta keto adipic acid is an intermediate, and while this is being formed, one molecule of coenzyme A and one carbon dioxide molecule is eliminated. So this is the step which leads to the formation of carbon dioxide. However, alpha amino beta keto adipic acid is an intermediate and it spontaneously changes into the final product of the first reaction, which is delta amino levulinic acid, and we'll mostly be using the abbreviation as delta ALA. So this is the first reaction, and you remember that this is the rate limiting step, and this is the step where the feedback inhibition also occurs, and this is the step where the delta ALA synthase enzyme in the liver is regulated. Always remember that the synthesis of porphyrins in the bone marrow or the erythropoietic tissue is always constant. The enzyme, so that means the enzyme in liver and in the bone marrow, the delta ALA synthase is different. One can be regulated and the other one cannot be regulated. So let's talk about the most important enzyme of porphyrin synthesis, which is the delta ALA synthase. Delta ALA synthase is the enzyme which is dependent upon pyridoxal phosphate for its synthesis. It is also the rate limiting step of the heme synthesis. Now this enzyme has two forms. It should be very clear to you, and you must remember that two forms from two different sources of where the porphyrin synthesis takes place: delta ALAS1 and delta ALAS2. The delta ALAS1 or the delta amino levulinic acid synthase 1 is the hepatic enzyme, whereas the ALAS2 is the erythroidal enzyme. And you know that hepatic enzyme is under regulation. So this hepatic enzyme is inhibited by its product, which is heme. And just to touch upon how heme inhibits the action or the porphyrin synthesis, is it represses gene transcription and it accelerates the mRNA degradation. So the mRNA forms for ALAS1 synthesis is degraded very fast, and it also delays the transport of the enzyme, the formed enzyme, into the mitochondria, because remember the first step occurs in mitochondria. So heme, which is the final product of the porphyrin synthesis, delays the transport of the enzyme back into the mitochondria. Since it is not there, it is not available for action. Glucose, that indicates hyperglycemia. Hypoglycemia, they induce the peroxisome proliferator activator gamma receptor coactivator 1, and they induce ALAS1 synthesis. So inhibited by heme and induced by glucose or hypoglycemia both. Hemin, which is the iron in the ferric form, it also inhibits ALAS1. Drug metabolism by cytochrome P450. So those patients who take in drugs which are going to be metabolized by cytochromes. So you remember that cytochromes are formed from heme. So if there will be more consumption of cytochrome, so we need more of heme synthesis, so they are going to induce delta ALAS1 synthesis. Whereas the other enzyme, delta ALAS2, is the erythroidal enzyme, and remember, again telling you, it is ubiquitously expressed, always constantly expressed. There is no feedback inhibition, and it corresponds to almost 20% of heme synthesis in our body. Let's talk about the hepatic control of the porphyrin synthesis. So we know in general the pathway: starting material succinyl-CoA and glycine, they undergo biosynthesis, finally form heme. This heme product it gives a negative feedback to the enzyme delta ALA synthase 1 and it inhibits the biosynthesis of heme. So this is the normal phenomenon. Once when the heme concentration decreases, so this negative feedback goes away and succinyl-CoA and glycine lead to the formation of heme. So that's in very simplest form how hepatic control of the rate limiting step of porphyrin synthesis takes place. But what happens when there is disease? When there is disease, so you have the various levels where the pathway is obstructed, where the pathway or the deficient enzyme is present, and beyond that, the pathway cannot proceed. So, in the initial steps of the pathway, where there is accumulation of delta aminolevulinic acid and porphobilinogen, so these are not complete porphyrins. They cannot react with light to form reactive substances. They are not affected by the ultraviolet light. So when such initial substances or the obstruction is in the initial pathway, that the accumulation of those intermediates leads to neurovisceral symptoms. So a patient might present to you with vomiting, with diarrhea, with abdominal pain, or a psychogenic symptoms. But when the pathway is obstructed in the later half of the synthesis, there leads to formation of porphyrins. So these porphyrins or the porphyrinogens, when they get exposed to the ultraviolet light, they react with the ultraviolet light and they form the free radicals in our body and they lead to the cutaneous symptoms or the photosensitivity symptoms. So this diagram I'm over here trying to explain the basis of the neurovisceral and the cutaneous symptoms that occur in the porphyria. Whereas when we talk about the erythroidal control, we must again remember, and I'm emphasizing that it is not under any regulation, it is constant and it is ubiquitously expressed. And the enzyme responsible for the erythroidal synthesis of porphyrins is your delta ALA synthase 2. So the second step of porphyrin synthesis, which is a condensation reaction, also occurs in the mitochondria. So from first step we had formed the delta aminolevulinic acid. So two molecules of delta aminolevulinic acid in presence of the enzyme delta ALA dehydratase. So it's a there will be removal of water. It's a dehydratase enzyme. This enzyme is zinc containing enzyme. So zinc is important for the porphyrin synthesis. And it leads to the formation of porphobilinogen. Short form for this that we'll be using will be PBG, and one molecule of water is eliminated. So this is the condensation reaction which is occurring in the mitochondria, step 2 of porphyrin synthesis. This reaction is inhibited by lead. So in cases of lead poisoning, when there is inhibition of porphyrin synthesis, that is the basis why a patient develops anemia when he is exposed to lead. Moving on to the synthesis of porphyrins in the cytoplasm. So step 3 occurs in the cytoplasm and it is a deamination reaction. The product from step 2, which is a porphobilinogen or PBG, moves from the mitochondria into the cytoplasm and is acted upon by the enzyme hydroxymethylbilane synthase. This enzyme has other names also. One of the names is porphobilinogen deaminase because it is going to cause deamination of porphobilinogen, and it is also called uroporphyrinogen 1 synthase. So in presence of hydroxymethylbilane synthase, 4 molecules of porphobilinogen combine. It's a condensation as well as a deamination reaction. So they combine to form the first linear tetrapyrrole. Remember, it's a linear tetrapyrrole and it is very unstable. So the first linear tetrapyrrole that is formed is known as hydroxymethylbilane, and this is the structure of the linear tetrapyrrole. So this is the step 3 of the deamination reaction. Now moving on to the step 4 of porphyrin synthesis, we are still in the cytoplasm and the next reaction that occurs is a decarboxylation reaction. There will be removal of carbon dioxide in this process. Before we go into the actual reaction, we must go through a few things, which are that at this point of time, two different pathways are followed. The hydroxymethylbilane that was formed in step 3 can undergo two different processes. First is the major pathway, in which it forms uroporphyrinogen 3, and the second is the minor pathway where it forms uroporphyrinogen 1. The difference between 1 and the 3 form is the arrangement of the carboxyethyl groups, that is the propionic acid, and the carboxymethyl groups, that is the acetic acid. The non-enzymatic conversion of uroporphyrinogen 1 results in the sequence AP, that is A is for acetate and P is for propionic acid. So ring 1 will have acetate propionic acid, 2 acetate propionic acid, 3 acetate propionic acid, and 4 acetate propionic acid side chains. Okay, so it is simple, it is symmetrical, there is no disharmony. Whereas the enzymatic conversion into the uroporphyrinogen 3 leads to reversal of one acetate and propionic acid group, which is in the last ring of the tetrapyrrole. So in the last arrangement of the tetrapyrrole, it is arranged in the reverse order. Instead of acetate and propionic acid, the arrangement is propionic acid and acetate. Coming on to step 4 of the porphyrin synthesis, we continue in the cytoplasm. So in the cytoplasm, the hydroxymethylbilane, which was our product from step 3, which is a linear tetrapyrrole, undergoes cyclization. So we know there is two pathways, either it will go in a minor pathway or it will go in a major pathway. So for minor pathway, we have the enzyme uroporphyrinogen 1 synthase. You know the other name for this enzyme is hydroxymethylbilane synthase and porphobilinogen deaminase. With the help of same enzyme, uroporphyrinogen 1 is formed, and this uroporphyrinogen 1, the four pyrrole rings are arranged symmetrically to each other, where the groups are alternated by acetate and propionic acid. In the major pathway, uroporphyrinogen 3 synthase leads to the formation of uroporphyrinogen 3, and in this you can see, like in the fourth cycle or the fourth ring of the pyrrole, the position of the side chains is reversed. Propionic acid instead in place of acetic acid and acetic in place of propionic acid. So this is the major pathway. So most of the hydroxymethylbilane goes into the major pathway and leads to the formation of uroporphyrinogen 3. Now these are the true porphyrins, uroporphyrinogen 1 and uroporphyrinogen 3. So whenever the patient is exposed to ultraviolet light, this inactive uroporphyrinogen 1 will get activated and will get converted to uroporphyrin 1, and it will cause photosensitivity symptom. Similarly, UV exposure for uroporphyrinogen 3 will lead to formation of uroporphyrin, and they both will lead to photosensitivity. Now during the normal metabolism, both these uroporphyrinogen 1 and uroporphyrinogen 3 are excreted in urine in some amount. Now going on to step 5 of the porphyrin synthesis. Step 5 is the last step of porphyrin synthesis that occurs in cytoplasm. It is a decarboxylation step. So in this, the substrate uroporphyrinogen 3, the structure of which is clear to you, it has four pyrrole rings. The fourth ring, the arrangement of propionic acid and acetate groups is reversed. So this is acted upon by the enzyme uroporphyrinogen 3 decarboxylase, and it gets converted into coproporphyrinogen 3 with elimination of 4 molecules of carbon dioxide. These 4 molecules of carbon dioxide are eliminated from all the 4 rings of the tetrapyrrole. The acetate in the tetrapyrrole ring is converted into a methyl group, leading to synthesis of coproporphyrinogen 3. This coproporphyrinogen 3, when exposed to ultraviolet light, gets converted into coproporphyrin, and it is responsible for the symptoms of photosensitivity in the porphyrias. Now this coproporphyrinogen 3 then finally moves into the mitochondria. Coming on to step number 6, which occurs in the mitochondria, again it's a second decarboxylation. In first decarboxylation, we removed 4 molecules of carbon dioxide, whereas in second decarboxylation, we are going to remove another 2 molecules of the molecule. So, coproporphyrinogen 3 comes back into the mitochondria and it is acted upon by the enzyme coproporphyrinogen 3 oxidase. Remember the structure? All the four pyrrole rings have now methyl group present at their positions instead of the acetate group. So it is acted upon by coproporphyrinogen 3 oxidase and is converted into protoporphyrinogen 9. Now, why it is called 9 is because at the time of discovery of porphyrins, it was the ninth compound to be discovered. So 9 has no clinical relevance. It's only during the discovery that it was named the ninth molecule. So coproporphyrinogen 3 acted upon by coproporphyrinogen 3 oxidase is converted into protoporphyrinogen 9 and 2 molecules of carbon dioxide are eliminated. So from where are these 2 molecules of carbon dioxide eliminated? They are eliminated from ring 1 and ring 2 of the tetrapyrrole, where the propionic acids are converted into the vinyl groups. So step 7, which occurs in the mitochondria, where you have the substrate protoporphyrinogen 9. So when you look at the structure, you can see that this is the ring 1, 2, 3 and 4. So methyl vinyl in ring 1 and 2, methyl and propenyl in ring 3, and in ring 4 this is propenyl and methyl. So protoporphyrinogen 9, which undergoes a reaction with the help of enzyme protoporphyrinogen oxidase to form the active form of protoporphyrinogen 9, which is known as protoporphyrin 9. And in this process it loses 6 hydrogens, and these hydrogens are lost from the tetrapyrrole rings, the internal bonds of the tetrapyrrole rings. Now coming on to step 8 of the porphyrin synthesis, which is the last step of the synthesis. This step takes place in mitochondria. We have the substrate protoporphyrin, which is acted upon by the enzyme heme synthase of ferrochelatase, which leads to the synthesis of heme. Now thing to remember over here is that for heme synthesis, iron should be available in the ferrous form. Ferric form of iron cannot be utilized in the formation of heme synthesis. So this is the diagram showing how iron forms coordinate bonds with the tetrapyrrole ring that leads to the synthesis of heme. So let's go ahead and revise the heme synthesis. The synthesis of porphyrin starts in the mitochondria. We have the substrates glycine and succinyl-CoA, which combine in presence of enzyme delta ALA synthase to form an intermediate alpha amino beta keto adipic acid, which is converted into the delta amino libulinic acid. 1 molecule of carbon dioxide and 1 molecule of coenzyme A are eliminated. Deficiency of this enzyme delta ALA synthase leads to sideroblastic anemia. Step 2. 2 molecules of delta ALA combine in presence of enzyme ALA dehydratase to form porphobilinogen. Deficiency of delta ALA dehydratase leads to DOS porphyria. Now porphobilinogen leaves the mitochondria and goes into the cytoplasm, where it is further acted upon. 4 molecules of porphobilinogen, they combine to form the first linear tetrapyrrole, which is hydroxymethylbilane. The enzyme for this reaction is porphobilinogen deaminase, and 4 molecules of ammonia are eliminated. The deficiency of porphobilinogen deaminase leads to acute intermittent porphyria. Now this hydroxymethylbilane can undergo 2 fates. Either it will follow a minor pathway or a major pathway. When it follows the minor pathway, it goes into the formation of uroporphyrinogen 1. And this uroporphyrinogen 1 is converted into coproporphyrinogen 1, and this constitutes the minor pathway of the porphyrin synthesis. The major amount of hydroxymethylbilane goes into the synthesis of the major pathway, which leads to the formation of uroporphyrinogen 3, and the enzyme for this is uroporphyrinogen 3 synthase. Deficiency of this enzyme leads to congenital erythropoietic porphyria. Then this uroporphyrinogen 3 is converted into coproporphyrinogen 3 with elimination of 4 molecules of carbon dioxide. The enzyme for this reaction is uroporphyrinogen decarboxylase, and deficiency of this enzyme causes porphyria cutanea tarda. Once coproporphyrinogen 3 is formed, it moves back into the mitochondria where further reactions takes place. This coproporphyrinogen loses 2 molecules of carbon dioxide to form protoporphyrinogen, and the enzyme for this reaction is coproporphyrinogen oxidase. Deficiency of this enzyme coproporphyrinogen leads to hereditary coproporphyria. Further, this protoporphyrinogen is converted into protoporphyrin 9 with the help of enzyme protoporphyrinogen oxidase, and the deficiency of this enzyme leads to variegate porphyria. And finally, the last step where protoporphyrin is converted into heme by incorporation of iron, which has to be present in the ferrous form. Remember, ferric form of iron cannot be incorporated into protoporphyrin 9 to form heme. So when heme formation has to take place, iron has to be in the ferrous form. The enzyme for this reaction is ferrochelatase or heme synthase. Deficiency of this enzyme leads to hereditary protoporphyria. Thank you for listening to my lecture. If you have any questions, you can email me at this address, which is bhavna.ghosh@yahoo.com, or you can WhatsApp me for queries. Or if you want to have one-to-one sessions with me, you can also WhatsApp me so I can discuss with you all the issues you have regarding this topic or any other topic in biochemistry. And stay tuned to my channel because I'll be bringing in more topics related to biochemistry for your study. Thank you so much.